Thank you. Thank you so much for inviting me and giving me opportunity to talk about my research. Of course, it's always a pleasure to be at Discoveries. And I learned that I have only 10 minutes, a little bit late, so I cut my title to a little bit shorter title. So I will focus on magic power and single carbon metabolism. This is what I like to explore, but I will tell you a little bit about dark part of life as well. So my first slide is very simple, and I just want to put everything in a concept. I want you to think about carbon cycle, yeah? What we know about carbon cycle, we have CO2, we have sun, we have plants to fix it, to bring it to biological materials. This is my interpretation of every organic compounds we can find in nature in a state. We found them in biology. And of course, then there is a decomposition of organic material, which is done mostly by respiration, which brings it back to CO2. And this is fast-running cycle on the planet. But there is also a slow-running cycle on the planet, which means that part of the carbon will be deposited as soil, sediment, and fossils. And this is my favorite definition of fossils and average. And they slowly will be released to become CO2 again. But of course, we as humans accelerate this cycle and we bring more CO2 to atmosphere. You know a lot about it already. What maybe not all of you know that there is another hidden part of this cycle, which is driven by serpentinization, which is completely a biotic process, which is driven by minerals in a mantle under high temperature. Water is hydrolyzed to produce hydrogen. It interacts with CO and CO2 to produce organic molecules, a biotic origin, and significant portion of these molecules is methane. And this is what I like to place my favorite part of single carbon metabolism, which is called methanotrophy, how biology can convert methane back to biomass, back to drive the carbon cycle. And of course, let me to move forward. Everything what you see so far, it was sun and it's driven. But now when we talk about this process, we talk about dark environment where there is no sun anymore. And the biggest question always will, to what extent Earth-derived carbon can support life on this planet? To what rate it contributes to it? And how has methanotrophy influenced Earth's climate in the past? And we know it's methane protected Earth when sun was cold. Now we know that it's warming up our planet. And how with this knowledge we can translate to mitigate some changes now. And people approach this many different ways. We have fantastic study coming from SIO, which demonstrates that right here at La Jolla, if you have animal spiders, the deep sea, about 15% of them will be fitted on this methane, which I found always exciting. But what excites me always, what biological properties enable methanotrophy, what it means to be methanotroph, what it means to live on a completely dark carbon. Now it's kind of, let me to pull everything on methanotrophy for you. Methanotrophy by definition is defined as ability to use methane as sole source of carbon and energy. It's some interplay between autotrophy and heterotrophy, because like autotrophs, they start with very often with abiotic carbon or single carbon compounds. They have to fix it. However, as heterotrophs, they still rely on organic compounds, so they still rely on respiration. And if I will put everything in a very short time frame, I would say methanotrophy require a very dedicated metabolic network, some of the most ancient pathway on the planet, like tetrahydromethanaptyrine pathway. Other parts more recently evolved. It will also require pathway which you can find across all domains of life. It will require UMP, which is very similar to glycolysis, which hardcore heterotrophs like us and E. coli. It will also have intermediate like glycine cycle, which you can find in plants, and of course CBB cycle, which you can find in plants as well for CO2 fixation. It's like one more microbe unites many different pathways, and I like to say that maybe this is metabolic is the LUCA, the last universal acceptors, which goes in many different ways how life extracts energy later on. Of course, methanotrophy, because it's partially autotrophy, relies on minerals, and it would be part of my talk today. And amongst those minerals are copper, iron, more typical ones, as well as less typical like rare earth elements, what we now call nowadays critical materials. So they extract them from nature. And what is another unique part, which I always am telling my students, when we look on our microbe, we have to remember their cell biology and cell structure. Because what I present you here, this is our microbe at one condition when it drives methane consumption, depends on copper. It forms membrane, almost like your mitochondria. It depends on respiration, packed with membrane inside. You remove energy domains, you move to another compound, and it will change the structure as well. And we find it across this unique cell biology, we will find across different phyla of metanotrophy. So that's a very short, fastest overview of metanotrophy I gave you. But again, what I want you to describe today is that metanotrophy depends on minerals, but it's not only conditions they utilize, that also minerals they have, and that will shape how they organize, and it will significantly shape what they express. What I show you here is the expression in response not to the carbon substrate, but the simple switch between copper and no copper condition, and I believe you might see here that respiration no copper will go significantly down. I will cover some of it at the end. Two stories I brought today, they're not complete, they're in preparation, and they're my biggest inspiration. One of them was completed by Dr. Marcus Bray, who worked in my lab. It was supported research by David Collins and Richard Hamilton, who were all postdoctoral students, now doctorals as well, and Merlin Thompson in collaboration with Rensselaer Polytechnic Institute and national labs. Very simple introduction to respiration. To respire, cell would need complex 1, complex 3, complex 4, and of course will be coupling. Proton will be pumped out, they will come in and produce ATP. Chemoosmotic coupling, Merkel theory in a very brief interpretation. What we found, what we were hunting for, and one big question in metanotrophy, what is the source of electrons to drive methane oxidation? We were searching for this for quite some years. We continue to search, but we decided to do experiments on membranes. We isolated those membranes, and we know that it's not an ADH. It's not just this one, something else between. And we did found that there is a cytochrome. We introduced cytochrome from outside, and cytochrome will be oxidized, and an ADH will be produced. That's what was expected. It's kind of partially is also known as a reverse electron transport. So we just to validate the study, we did a number on inhibition experiments. Inhibition, I believe we all know, if you add cyanide, somebody will die because the terminal oxidases will be eliminated. So we just did it inhibition for each individual step, and what we found was somewhat surprising. If we add cyanide, it drops. We add ATP back, and after that, as soon as cell has ATP and reduced cytochrome, it always will produce an ADH. We were surprised to a certain point because it seems to be even if we disturb completely proton gradient, it still will produce an ADH, and it's still respiration machinery, and just does not fit into theory. So we went to our colleagues and we asked, do we really observe an ADH, our experiments? And they did, in my opinion, another beautiful study with fluorescent lifetime imaging microscopy, which can detect fluorescence directly from an ADH. Only an ADH, it can't see an AD. What they took our membranes and they put them under microscopy and they investigated the same. What they found, and that was surprise number one. After all purification steps, membranes were saturated with an ADH, and this NADH was bound. As soon as we added cytochrome, reduced cytochrome, we see an ADH production even more. It lights up. I hope you will see this lights up. Again, potassium cyanide does not impact it, and any disturbance in proton gradient does not distract it as well. This is a mystery which I'm summarizing here because, again, we have several surprises, like membrane has capacity to hold a lot of redox even after several steps of purification. But the main point I wanted to say is that we now know that there is existence of membrane-bound machinery which can take cytochromes and just ATP, most likely from cytosol, to produce an ADH. This is, I will not say completely contradict Mitchell coupling theory, but it's at least tell us that there is another way how we in cells, like mitochondria, can recover energy, and we still don't know exactly what is happening there. My second favorite story is not complete, and I brought even my student who's still working on this one, but I wanted to show you another element which I like a lot. Recently, we were funded by Department of Energy to investigate if we can use our microbes to recover critical material. As we look on pathways for recovery of critical material, we look on a number of different minerals, and we looked on copper as one of them, and what we found that and we also place cells under severe metal limitation. And what we found under severe metal limitations that actually we see those small extracellular vesicles. And we decided to follow up what it means. I show you another image of this, and this image exactly the same population, but now this population lives under metal restriction. What you will see, this would be a very healthy cells, but now I want you to follow from this cell to this cell to this cell and that. Basically have formation of extracellular vesicles in one shot. What we did after, of course, we have formation. We can even look inside of cells for a minute. We can separate extracellular vesicles, and we can perform simple experiment to see if this extracellular vesicle can help extract minerals. So what we did, we put those inside of the tube, so it's separated, cell cannot travel, and we place cells outside. This is cell culture, and we look if cells can grow or cannot grow. And what I believe you all will agree is that we see more growths on this side. So we calculated growth only cells in which minerals, and it was red mud, high pH minerals, and it's high pH environment. What we were able to observe that extracellular vesicle accelerated metal recovery. We even were able to measure those in cells, suggesting that metanotrophic bacteria can enable a recovery of minerals, but they use very, in my opinion, unique pathway for that. We calculated, this was done by China Baum, and I asked her to sit down and calculate single cell in the population. And what we found that about 70% were normal cells which continue to be a single cell, about 15% continue to divide, and some portion of cells decide to go into granulation pathways, our so-called apoptosis, our programmed cell death. And this apoptosis leads to formation vesicles, which then help to extract minerals from otherwise insoluble, which then support population, other members of population to grow. So in summary, I want to say that that was a surprising to see this truism in microbes, our cell differentiation. We would love to explore it further. But on top of that, I believe I wanted to comment with what we see also here is the apoptosis, which in other organisms typically linked to toxicity, to redox. I believe we have to look at it in slightly different eye and look on mineral limitation in driven apoptosis. I don't think it was touched base before. And with that, I'm not sure how I'm doing on time, but I would love to say thank you to all my current members and people who contribute in many different ways, like Will Gebbe, who performed a number of analyses, as well as the former members who contributed to some aspect of this project in many different ways. We have a number of collaborations. And despite me being stubborn and sometimes very tough on my team, I still believe I have best team at SDSU. And with that, I'm happy to answer your questions. Okay.
So for the EVs, for the extracellular vesicles, I saw, I think it was like a table kind of showing, I think it was rare earth elements, iron, copper being recovered.
Yes.
Okay, yeah, I just wanted to say.
Calcium,
sorry.
Yes.
Calcium. So it was iron that was recovered.
The iron was recovered the most, and this is the most which is in cells. The cell demand number one is iron. They will hunt for iron. Then calcium, this is a key enzyme, and if calcium is limited, they also can switch to rare earth elements and extract them as well. It also partially reflects what is in, let's say you don't see copper because there are not much copper in red mud, but what it does reflect that we have over presentation of lanthanides compared to its natural abundances in the minerals. So it's not only recovering, solubilizing minerals, it's also specifically, this is why I put chelating molecules, and this is why we have to hunt for those, because I feel like in addition to discovery in fundamental research, we have very nice opportunity to build a technology which can help with taking minerals from tough materials. Okay. Yes?
So you mentioned, these different critical minerals, and I see that that's doing things differently for the different minerals. Is there a way, or is there an advantage to, and is there a way to possibly like program or alter some sort of genome to seek out specific
fantastic question. I believe you just follow the logic of engineer. If I would be, say, what would be next step? You identify this apoptosis pathway. What next you want? You want to grow cells. You know how to induce apoptosis. You convert all your cells in these mineral extracted granules. You also dissect everything to know which exactly compound is needed to extract different mineral, and you definitely follow that, and then you have only one set, you restrict everything else, and you can restrict just one mineral. That's, I mean, the only part which most investors would like, they would like to do it, have it next year. So I have to say most likely not next year. So there is quite a bit of fundamental research which still needs to be done to unfold all elements and interplays. We're just starting it, okay? Oh, good. So we got almost the same time.
So I don't have a question, but I have an invitation to you to take us to the future.
Okay.
Say 10 years. I'm going to give you 10 years. How do we scale this? How do we scale this up? And what benefits do you envision?
So how we scale this up and what benefits I will envision for that if we will go and scale up, yeah? Number one is that metantrophic bacteria, especially the one which I showed you here, is microbe which can grow on brackish water like seawater. You don't really interfere with other parts, yeah? We can grow this microbe, and we tested it at scale. We know that we can bring microbe to scale at which the cost of production per ton of element would be around 1,000, which is the cheapest price you can find for production of microbes, because we can use biogas. That's another sustainability loop which you can add here. Now the second part, if you dissect everything and you know those chelating molecules which specifically separate from red mud, we have humongous amount of red mud here in US, and it's actually environmental concern because it's soil contamination. So even if you're not really pushing, I want to do mineral extraction because I think that scale for mineral extraction is somewhere else. But I do want to do remediation of soil in the most sustainable way. I feel that moving forward might bring it very close to you because red mud is high pH. We have high pH microbe. Proteins from this microbe are already adapted to high pH environment. So exploring it definitely can bring benefits for removing at least toxic compounds or some contamination on sites. Extracting it at the scale that still will be feasible, there is a high push to that, and I have to say that there is a strong interest. We still have revision, different things how we approach this particular, because the scale is different, guys. When you look on mining, the scale of mining does not really fit immediately in this biology, and as why we are right now, so justifying biology with fermentation because it costs. We always have to come to sustainability with the idea that everything is cost. Producing cell is a cost, not only money, it's energy and CO2 input. So in certain areas it's feasible, it's not translatable to completely everything. I don't envision that we will completely replace mining this way. I do envision that we can replace mining, but coming up with completely different polymers to do the job. And again, invite me in 5 years to present on other ways of storing energy and doing that. Okay?
Thank you so much.
Thank you. I will answer your question later, I believe. Yeah.